Hello everyone. It's been long. It's been long we uploaded any video, but I'm back. I'm back with my series of lectures in general biology, biotech, forensic science, etc. So this morning, what are we going to discuss? This morning we have an interesting topic in the field of forensic biology. So we are going to learn how to identify human remains using DNA technology. And this is often applied when serious crimes are committed like murder and the body is concealed and later found. In such cases, only human remains are discovered. So, when you come, don't forget to subscribe to our channel for us. Also, like, comment, and share this video. So, just go ahead and subscribe for us. I'm waiting for you. Subscribe and let's move on with this lecture. Okay. So, I hope you've all done that. I hope you've all subscribed to this channel. So, we can move on. So, introduction. You say the body of a deceased person in whole or in parts, regardless of its stage of decomposition. That is human remain. And human remains may be encountered as a either recently dead body, any stage of decomposition, bent or mutilated remains, and also skeletonized form, like the bones, teeth, nails, whether fingernails or toenails. So human remain fall under these four categories. When do we encounter human remains? You can encounter human remains in serious crimes like murder, in plane crashes, disasters, fire, or war. And why must we identify these remains in the first place? Someone will say, of course, these are remains. So why must we spend so much money in identifying them? One is to bring relief to the bereaved family. It is not easy waking up every day and thinking that your beloved might be somewhere, just that you cannot find the person. So when identification is done and you get to know that in reality you've lost your beloved one, then you can mourn the dead and move on with your life. Also, it is important to identify a human remain so the remain can be given a befitting burial. Another one, so justice can be obtained and the perpetrators sent to jail. Some forms of identification. One is the physical examination, using the jewelries, the marks, and tattoos, etc. of the deceased person. That's if the body has not been skeletonized. Or even if the body has been skeletonized and something like jewelries can be found, it is also a form of identification. Fingerprint. is also another form of identification of a deceased body. Odontology is also another field that can be applied in identifying a human remain. And the modern one, DNA, that using nuclear DNA, mitochondrial DNA, or you can do Y chromosome analysis when the supposed deceased person is a male. So once again, please don't forget to subscribe to this channel. Also like, comment, and share this video for us. So please go ahead and subscribe for us before we continue this lecture. I hope you are doing so. I hope you've subscribed to this channel. So let's move on. Thank you. Now, some specialists involved in human remains identification. We can employ the services of these individuals or these specialists in identifying a human remain. One is an archaeologist. Two, an anthropologist. An entomologist. A medical examiner. An odontologist. Fingerprint expert. And also a DNA expert. And what do we do or what to do before sampling biological samples for human identification? Or sampling anything, even if not biological material, for human identification. When you find the deceased body, what do you do before you take anything that you deem it fit for identification? First thing to do is to interview people, and this is to gather and verify information. You do examination, that you examine and evaluate the scene where the body has been found. Photograph. You take a photograph of the scene. You also sketch the scene. Then you can go ahead and process the scene and then take your samples. So it's just like the normal crime scene processing procedure. Recovery and sampling. How do we get evidence and reference samples when it comes to identification using DNA? So we are now limiting ourselves to our topic of interest. That's identify human remains using DNA technology. So from here, we are going to limit ourselves with DNA to DNA technology. So we are going to identify human remains using DNA technology. We have two types of sample. We have what we call the evidence sample, that is the unknown. If you find a body that has been decomposed beyond recognition, taking a sample from such body for human identification involves taking the evidence sample and the reference sample. The evidence sample is what you are taking from this decomposed body, and the reference sample is what you are going to compare it, either from relatives, or you are going to compare it to a sample the deceased person left behind, a known sample from the deceased person. So how do we obtain evidence sample? Through direct recovery from the scene, and also through exhumation. How do we get reference sample for comparison? Through direct sample from victim, that is a sample the victim or the deceased person left behind. Then also sampling from relatives, especially close relatives like the father, the mother, the children. If these samples are not available, then you can fall on longer distance relatives like siblings, the grandparents, etc. But then normally close relatives are preferred. Then if you are lucky to have a DNA database, after profiling the evidence sample, you can as well search through this DNA database if you get a hit or a match. So DNA analysis. You can use the nuclear DNA, which is the most preferred form of DNA when it comes to human identification. But if this type of DNA, after extraction, quantification, you get to know it has degraded, then you can fall on what we call mitochondrial DNA, which is normally available. You can also do what we call Y chromosome analysis if the individual is a male. So what are the steps in this DNA profiling process? That is what every scientist or a DNA expert goes through before getting a DNA profile. You prepare the sample first. The sample preparation depends on the type of sample you have, whether bone, nail, flesh, blood, etc. Then you can go ahead and extract your DNA using an appropriate kit. There are a whole lot of DNA extraction kits on the market. After extracting your DNA, you have to quantify the DNA. What do we mean by quantification? That is knowing the amount of DNA that you have extracted. It can happen that you didn't even extract anything at all. So it is very, very important that after extracting your DNA, you quantify to see the amount you got. With the introduction of modern quantification kits, you can even get to know whether your sample has been contaminated. You can get to know whether your sample has been degraded. You can get to know whether you extracted DNA in the first place or not. You can also get to know whether you have PCR inhibitors in your DNA sample. So quantification of your extracted DNA will give you a clue before you even proceed to the next step, which is the PCR. So your next step is the PCR or polymerase chain reaction. You can either analyze a single locus or you can do a series of locuses at the same time, that's multiplexing. Case like global filer, identifiler, the Promega case, they all allow multiplexing, which is very good in forensic. After doing your PCR, you go ahead with electrophoresis. And after this electrophoresis, you can then get to know your DNA profile. So these are the steps in the DNA profiling process. After getting your profile, then you come to profile comparison and the statistical calculations. If you have a direct sample to compare it to evidence sample, then you should be getting 100% match. It means the DNA profile that you generated from your evidence sample should match exactly. They should be the same as your reference sample if the reference sample is coming from the deceased individual. If they match, then you can conclude that the individual in question or the deceased person is the person that you suspect. If you are going to compare your evidence sample to a sample from a parent or child of the deceased individual, then you should get a 50% match. It means at each single locus, the deceased person should have an allele matching with an allele from the individual you are comparing with. That is the 50% match. Then what conclusions can you draw from such analysis? One, you can exclude the individual as being the person that you are suspecting to be. Then you can also include the individual as the person you are suspecting to be. Then depending on your sample, especially if you have degraded sample and you do nuclear DNA analysis, your results can be inconclusive because of what we call allele dropouts. You can drop some of your alleles. Or if you're not lucky, in some situations, your sample is contaminated and becomes a serious form of mixed sample, then calculations become difficult. So there are a whole lot of scenarios that can lead to inconclusive results. So let's look at a case study where a human remain was identified using DNA technology. So on the 7th of August 2012, a woman went missing. Then on the 15th, a suspect was arrested by the police. Based on interrogation, crime scene management team discovers disturbed area on the missing woman's farm. Drops of suspected human blood leads to suspect farm. So, investigators pick blood that has been mixed with soil from the suspect farm. That's suspected blood sample. Then, this same crime scene management team discovered disturbed area on suspect farm on the 20th of September 2012. The removal of the soft soil leads to a foul smell. So they invited a pathologist and a disfigured body was exhumed. So the body was disfigured. There was no way any person was going to recognize the person through facial recognition. So once again, my viewers, please don't forget to subscribe for me. Also like, leave your comment and share this video. So let's take 30 seconds to do this. Let's subscribe. 30 seconds. Let's do so. 30 seconds. Please subscribe so we can continue. Please subscribe. Thank you. I hope you've all done so. So let's continue. So the exhumed body was sent to the morgue, where a bone sample was also taken or sampled by the pathologist and sent to the forensic lab. Also, the soil blood mix sample was also sent to the forensic lab. After doing serological analysis, a soil mix suspected blood sample tested positive for human blood. So first they use what we call the hemastis test, which is a presumptive test for blood, and they got a positive result. After which OBTI, hexagon OBTI test, was used to confirm the presence of human blood. And it was confirmed that indeed the soil mixed suspected blood sample is blood. So human blood was mixed with soil. Then buccal swabs of the son and mother of the missing woman, was taken or were taken. So sampled from both the son and the mother of this missing woman. So 3 samples were used for DNA profile. One, the bone sampled by the pathologist. This blood that has been mixed with soil, and then the buccal or cheek swab taken from the son and mother of the missing one, in this case, the reference samples. So the samples were taken through the stages that we already talked about: DNA extraction, quantification, PCR, electrophoresis, and then what did they get? They got a full DNA profile for the bone. It means they got complete alleles at each locus that they tested. There was no degradation. Then for the soil made blood sample, they got few alleles. It means the sample had undergone some form of degradation and there were allele dropouts. For the buccal swabs or the reference sample, they got full profiles for both the alleged son and the alleged mother of the missing woman. What form of analysis did the experts make? Now they conducted a maternity test, a reverse maternity test. So if you look at this, if you look at the slide, these are what we call the various loci. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 different loci, plus the sex determining locus, or what we call the amelogenin. Short form here, amel. That is, if the person or if the sample that you are processing is from a male or a female source. So if we look at the first locus, this is the DNA profile from the skeletal remain using the bone. And this is the supposed mother of the source of the skeletal remain. So you could see that they are sharing allele at this particular locus, 13, 13. If you go to the next locus, 30.2, there was 30.2 here. So using the principle of maternity test, they got to know that the supposed mother and this skeletal remains share alleles at every single locus tested. So maternity index were calculated and there was a combined maternity index. The experts went ahead and did say between a child, that the supposed child of the source of the skeletal remain, and once again, the supposed child and the source of the skeletal remain shared allele at each locus. So maternity indices were calculated. So what statements were made by these experts? They concluded that the alleged relationships cannot be excluded. That is, what they were suspecting, that the sample that they got from the crime scene could be that of the missing woman. They were able to establish that using DNA technology. The combined maternity indices for the two analyses, that for the supposed mother and skeletal remain and the supposed child and the skeletal remain, were 69250 and 20876 using the appropriate allele frequencies. The probabilities they obtained were 99.999% and 99.995%, which is acceptable by international standards. So we come to the end of today's lecture or presentation. I'm very grateful for watching. Thank you very much, and I hope that you've learned something from this. That's how to identify a human remain or a skeletal remain using DNA technology. If you have any questions, please do well to leave them at the comment section. I'll answer every bit of your question. So leave them in the comment section and I'll attend to your questions. Thank you. And also, don't forget to subscribe, like, comment, and share this video so others can also benefit from it. Once again, let's give ourselves 20 seconds for each and every one of us to subscribe to this channel. So we are counting 20. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Thank you very much for your patience. I hope you've subscribed to this channel. I'm very grateful for that. So look out or watch out for our next presentation or lecture. That is how to extract DNA from bone samples. A very interesting topic. Bone is a hard tissue, so how do we extract DNA from bone? Coming up, so watch out. Thank you very much and do have a very good day.